So today we'll start with kingdom fungi. So in last class we saw about kingdom protista and the various groups of the kingdom protista. So today we'll deal about fungi, the important characteristics of fungi and its modes of reproduction. So before that, if you didn't subscribe our channel, please do subscribe and press the bell icon for further updates. So we'll move on to the session. So first is kingdom fungi. Alright, now the first characteristic of kingdom fungi is that they are all heterotrophs. So none of the organisms in this group are autotroph. Alright, so they depend upon other organisms for its food and metabolism. Alright, so first is saprophytes, which means that the organism is relying on a dead organic matter. You call it as saprophytes. Example, mushroom. Next is parasite. So they depend upon a live organism, a living organism. So when it is depending upon a living organism, you call it as parasite. When it is depending upon a dead organism, you refer it as saprophytes. Next is symbiont. This is a very important characteristic of the kingdom fungi, where you have two important symbiotic phenomena. The one is mycorrhiza and the other is lichens. In mycorrhiza, when the fungi as in symbiotic association with the roots of higher plants. There's a plant and there's a root of a plant under the soil. Now what happens? These fungi, they dwell under the soil close by the plant's roots. Alright. Next is lichens. So when they are in symbiotic association with algae. So there's a group of algae, inside which there's a group of fungi. So there's symbionts. So which means that you call it as there is no harm between these two groups. Alright. So now what is very important about this symbiotic means of nutrition is that both are benefited. For example, I told you this mycorrhiza where there is a plant root and around the surface of the root there are fungi which has been dwelling. Now what happens? These fungi, they get its food from the plant's root and they use it for their metabolism. In turn, what they give is that they help the roots of these plants in a proper uptake of water. So they increase the porosity of the soil, so thereby the roots can easily suck water from the ground. Alright, so that is symbiosis. Both are benefited. Next is lichens. The same way in algae, what happens is that these fungi, they give protection to the algae. In turn, they get food from algae. Alright, so both are benefited. There is no loss, there is no gain. Both are mutually benefited, and you call them as mutualistic relationship between two organisms, and that is symbiosis. Alright, so we saw about what the nutrition in fungi. Next, we'll talk about the reproduction in fungi. So fungi undergoes vegetative reproduction, that is asexual reproduction, and sexual reproduction, which involves gametes. Now in vegetative reproduction, we spoke about budding. Example, yeast, a single cell yeast and there is a bud. Now what happens? Unidentical daughter cell. One is much larger, bigger than the other daughter cell. So that is what is budding. Next is spore formation. There are n different number of spores which is formed in fungi. You have sporangiospores, zoospores, actinospores and gonidiospores. Now zoospores and actinospores we saw earlier. So when the spore is motile, which has flagella, you call it as zoospores. When actinospores are those which are non-motile, they don't have the organ of motility, that is the flagella. Now we will talk about sporangiosspores and gonidiospores. When these spores are formed inside a sac-like structure, so this is spore and this is a sac-like structure, you call it as sporangiospores. Next is gonidiospore, where there is no sac, only the spore which is naked, you call it as gonidiospore. Alright, gonidiospore, there is no sac. Sporangiospore, there is sac. Zoospore, motile spores. Aplanospores, non-motile spores. Next, we'll talk about the sexual reproduction in fungi. Now, the sexual reproduction in fungi happens in 3 steps. Now, first step is plasmogamy. Alright? So, gamy means fusion. Now, what is fusing? The protoplasm of the cell. The cell could be a motile cell or a non-motile cell. What happens? The protoplasm of both of the cell, they fuse together, and this process is called as plasmogamy. Now, this plasmogamy is followed by karyogamy. So, gamy is fusion. Now, karyon is the fusion of nucleus. So now what happens? Apart from the nucleus, all the other cellular content is being fused. Now next step is the fusion of nucleus, and you call it as karyogamy. Now what happens is that in karyogamy, now 2 haploid spores, they fuse together and now they form zygote. Alright. Now always zygote is diploid cell, which is 2n in number. Now this zygote will undergo reduction division, meiotic division, and now they give rise to haploid spores. Now these haploid spores will develop into a new organelle, a new fungi. Alright. So now in this fungi, people usually call it as fungi and fungi, but I prefer to call it as fungi. I think that's a proper pronunciation. Next is dikaryon. Now what happens? In few groups like ascomycetes or basidiomycetes, they undergo something called as dikaryon stage, which means that the parents, see, first in karyogamy, what is happening? The parents are always haploid. One end, the other end, they fuse together to form 2n, diploid. But in case of this dikaryon, both the parents are 2n in number. Parent A, diploid parent. Parent B is diploid parent. Now they are fusing together. Now what would be the zygote? Alright. So now to reduce this 4n number of the zygote, what happens? These parental nuclei, so I mean to say not the cell, they have 2 nucleus. Alright. See. Parent A, which has 2 nucleus, 2n. Next, parent B has 2 nucleus. Right? Now, when they fuse together, what will happen? You have 4 nucleus. Clear? But now what happens? This can never happen. This should actually not happen. But this happens in ascomycetes and basidiomycetes. So now what happens is that the parent nuclei, they fuse together and they form 2n. Now clear? 2n and the zygote eventually becomes 2n because the out of the 4 nuclei, the parent nuclei will fuse together. So that is called as the dikaryon stage. Dikaryon, 2 karyon nucleus. So 2 nuclear stage. Alright. So in today's class we spoke about kingdom fungi and the characteristic features of kingdom fungi, then the sexual reproduction in fungi. So in this class we will deal about the different groups of fungi. So before that we will see few more important characteristics of fungi. The first is a fungal cell could be a unicellular, which means single cell, or multicellular, which means many cell. For single cell, yeast, and for many multicellular you have normal mushrooms. Alright, and now next you have to, based on their cell body, which is hyphae, you have two important things: septate hyphae and aseptate hyphae. So septate hyphae is that when each of these hyphal cells is been differentiated with the help of a septum, you call it as a septate hyphae. Now in other terms, all of the cells are been blocked. There are different cells which are differentiated, right? Single cells. And now when these hyphal cells are not separated by septa, you call it as aseptate cells or choanocytic cells. So this you would have studied in your lower grade. So this is all about your hyphae, septate hyphae, aseptate hyphae. And now we have four important groups of our fungi, right? So phycomycetes, ascomycetes, basidiomycetes and deuteromycetes. So you can remember it as BPAD. Alright, so basidiomycetes, phycomycetes, ascomycetes and deuteromycetes, BPAD. Okay, now in today's class we'll deal about this phycomycetes, and next class we'll see the rest of the groups. Now phycomycetes. So mostly the organisms under phycomycetes are aquatic fungi, and they are all the parasites of aquatic creatures, right? And now they have choanocytic hyphae, which means that there's no septa between the hyphae, there's no septae formation. That is choanocytic. And our reproduction is only by asexual means. So they have motile spores called zoospores, and they have non-motile spores called as aplanospores. And now a typical example of this phycomycetes group is mucor, rhizopus and albigo. And now based on the gametes, so the asexual gametes is isogametes and anisogametes. So when the gametes are similar and it is difficult to identify between the gamete, you call it as isogametes. Now both are similar. Anisogametes is that one could be a motile gamete and the other could be a non-motile gamete, or one could be little smaller than the other gamete. So that is isogamete and anisogamete, and you call it as heterogamete. Alright, so anisogamy is also called as heterogamy. So in today's class we spoke about the two different types of hyphae in fungi and the four important groups, and we dealt briefly about phycomycetes. And next class we'll deal about the other groups in fungi. If you want a screenshot, you can take it. Update. So we'll move on to the session. First is ascomycetes. And now this fungi, you call it as sac fungi because they resemble a sac, right? A spherical body this way, and that's why they are called as sac fungi. Next is basidiomycetes. You call it as bracket fungi. It's all because of its shape. All the name, as it says, it's all because of the shape. Next is deuteromycetes. It's called imperfect fungi because the asexual stage of a fungi is placed in some other group and the sexual stage of the fungi is placed in some other group. Alright, the fungi as such is not placed in a particular group of deuteromycetes. So we'll see all this clearly one by one. First is ascomycetes. So as I told you, it's sac fungi. It is also called as sac fungi. And a predominant characteristic of this fungi is called as they exhibit coprophilous type of feeding. So usually you call it as coprophagy. So copro, the term copro means feeding upon the fecal matter, dung. Alright, so feeding on the dung is called as coprophagy. Alright, so when you when you take these rabbits, they have this habit of feeding on their own overnight feces, and you call it as coprophagy because they eat their own fecal matter, overnight fecal matter, their own dung. So that's called as coprophagy. So the same way, this fungi are coprophilous. They grow on the dung and they feed on the dung, use the organic matter and then for their own metabolism, right? So that is called as coprophilous, which means they grow on dung. Next is reproduction is through spores. Two types of spores, conidiospores and ascospores. So ascospores, as I told you earlier, they are all non-motile spores, and this is the sexual part. In asexual part, you have this conidiospores. So example, you have this aspergillus, agaricus. So agaricus is what? Are edible mushroom, but not all the mushrooms are edible. Mushrooms like agaricus bisporus, agaricus. There are different types of species of mushrooms. You have button mushrooms, ring mushrooms, little elongated mushrooms. So a few of the mushrooms are edible, the other few are, they produce toxins, right? And next, and mostly the colorful mushrooms like pink color, orange color, they produce high amount of toxins, right? They are all poisonous mushrooms. Once when you eat it you will end up in getting toxins accumulated in your body, like food toxins, right? So that is this ascomycetes. Next we'll talk about basidiomycetes. So you call it as bracket fungi, and they're all parasites on plant and animals. So they don't have sex organs. So in 3 stages of its sexual life cycle, I told you earlier in my general characteristics of fungi video, which is a dikaryotic stage, which means 2 nuclear stage. So that's how they undergo their reproduction. And now the fruiting body is called as basidiocarp, and the plant body is called as basidia. So for example, the plant body, which means the asexual reproduction body, a vegetative part, is called as basidia, and the part which undergoes reproduction is called as a fruiting body, and that is basidiocarp. Next is deuteromycete. You call it as imperfect fungi. I told you, right? It's why? Because the vegetative part of this fungi is placed under its own group, deuteromycetes. But the sexual part of this fungi is discovered later. After this fungi is placed in deuteromycetes, the next sexual part was discovered later. So it was kept placed in this group, ascomycetes group, the sexual part. But the asexual part, the vegetative part, is placed in this deuteromycetes group. So that's why you call it as imperfect fungi. And common examples are Alternaria and Colletotrichum. So next class, we'll deal about virus, viroids and prions. So that's it for today. So if you want to take a screenshot of it, you can take. Now, first of all, in the 5 kingdom system of classification by Whittaker, he didn't place virus in a separate kingdom. Do you know why? It's all because viruses are considered as non-living organisms. It's because they are all crystalline form. Only when they attack a host, when they attack humans or animals or plants, they can actually multiply. Once they are all on the ground, they are all non-motile or mobile. They cannot do anything. They cannot undergo their own metabolic processes. They are considered as non-living things. So that's why Whittaker didn't place this viruses into a separate kingdom called kingdom virus. So apart from this 5 kingdom system of classification, the excluded one is viruses. So they are all a separate category. They are not into any kingdom. So we are going to talk about viruses today. Now first, virus. Now, discovery of virus, it was in the 18th century by Mitri. So, this exact how did Mitri Ivanovsky discover virus? I have a separate video on the discovery of virus, and I'll mention the link below in the description. You can go and view it. So now we'll talk about the characteristics of viruses, how exactly this virus look like. And our first thing is any virus will have a coat. So you will, in case of bacteria, you will say they have a cell wall, right? But in case of viruses, you will say that they have a protein coat. Alright, and now this protein coat, you call it as a capsid, which means the outer covering of the virus. Next, this capsid is made up of monomer of proteins, a smaller protein subunits, and you call it as capsomeres. So, capsomeres together form capsid. Alright. Next, inside the virus, you have the genetic material. Now, genetic material could be RNA or DNA, ribonucleic acid or deoxyribonucleic acid. Now, what exactly is this RNA and DNA is that any organelle should have a genetic material. Alright, do you all accept? Any organelle should have a genetic material. So in case of viruses, it is not exactly the DNA or RNA. Each of this virus could have a single strand RNA or a double strand RNA. And in case of DNA, it could have a double stranded DNA, but no way the single stranded DNA. So that is all about the nucleic matter of a virus. Now when you talk about the shapes, virus have n number of shapes. It could be helical, for example, tobacco mosaic virus, cucumber mosaic virus, and it could be a polyhedral or icosahedral or simply a spherical virus. For example, the virus which affects the body in terms of a common flu cold, they are just a spherical virus. Alright, so now the cold common flu itself is caused by virus, normal common flu virus. So this is all about virus, and in virus you have a separate subcategory called as bacteriophage, which means virus which attacks bacteria, right? So you know virus will affect plant, animals and humans, but there are separate group of viruses which will attack which will attack the bacteria, and that you call it as a phage, bacteriophage, viruses which infect bacteria. And this is because of two important life cycle, which is lytic cycle and lysogenic cycle, and these two cycle I'll explain it in the future classes. Alright, so for now you can remember lytic cycle and lysogenic cycles. Now this is all about virus. Next is about viroids. Now later, first how did they discover virus? Okay, bacteria. Virus is much more smaller than the bacteria, so that's why they call it as viruses. So if you see my previous video, you will get to know the discovery of viruses. The same way, now virus they consider as a very smaller organelle. Now what happened was, in case of a mad cow disease, what happened, they found few organelle, a virus which is much more smaller, right? And now they termed as viroids. Why they termed as viroids is all because they are derived from viruses. Only thing is, it does not have a shape or size or a proper structure. They don't have a protein coat or a capsomere. They just have a genetic material. It could be RNA or DNA, and they infect animals and plants, and they are much smaller than the viruses. So that's why they are placed under a separate group called as viroids. Next we'll talk about prions. So P for protein. So you can remember prions. Now what happens? A protein has to fold, right, during the time of DNA replication, during the time of replication, the post-replicatory process is that there has to be proteins which are synthesized. In the formation of synthesis of proteins, what exactly happens is that the protein has to fold. So there are different types, right? You have alpha helical structure, beta sheet structure. Now this helical and the beta sheets, they have an abnormal folding, and now this abnormal protein, they tend to be like toxins to the organisms. So this was first isolated in a person who got this Creutzfeldt-Jakob disease. It is almost like a Alzheimer come a nervous disorder disease, where you have shivering of hands as well as you have memory loss, right? So that is what called as Creutzfeldt-Jakob disease. But this is not exactly a non-communicable, neither a communicable nor a non-communicable disease, because these prions are present in the blood. So this happens only the blood of an infected person is being transfused to a healthy person. So the saliva of a person, like they can never orally transmit the disease, right? It's only in the blood, and even if it is transmitted through blood, is still a question mark and a fact of research today. And yes, prions are smaller than viruses. Viroids are much smaller than the viruses. Alright, so in today's class, we spoke about virus, prions and viroids. In next class, we'll start with a new chapter. If you want to take a screenshot, you can take it.